And now Bartolomeo, Celli, you can't complain. We didn't keep you busy.
No. I've been very busy. Can you get it on?
Combination
Well,
therapy.
I want to thank you for being busy. I've enjoyed very much the visit. I enjoyed very much going to Ferrari. And I'll be rooting for El Cavallino Rampante next year, too. So, so here we go. My conflict of interest are shown here. I must say that I envy the cardiologists, and I wish I were them. Our data will not be as solid. It will be a little more drafted by a personal opinion than it will be by evidence, supported by evidence. So here is what I would like to prove to you, that if we test COPD patients acutely on bronchodilators, they respond to bronchodilators. We'll then go into therapy with bronchodilators, then introduce the anti-inflammatories, pose some dilemmas that will be practical and theoretical, and then perhaps give you a Bartolome logical approach. It will be a little biased. So let me try to prove first that COPD patients respond to bronchodilators. And for that, I am using data from Uplift. This is a good study because it was large. It was 6,000 patients, and they had a mean FEV1 of 1.1 liters, around 45% of predicted. Cardiologists, 45% EF. A little low, somewhat compromised. Now, this study had one big virtue. We gave them two lonely, short-acting bronchodilators, Atrovent and albuterol at maximal doses, and we monitored what the response was after 1 hour of those two bronchodilators. I want you to see two things. In spite of having 1 liter average, 1.1 liters, 53% of patients had more than 200 milliliters response, a 16 to 20% response in bronchodilators. But the second fact that I want you to see is on the left side of the scale that there was a 7% of patients who actually constricted between 200 and 300 milliliters. Now, when I go to the additive or non-additive effect of two bronchodilators, I want you to notice this because I think this tells me that if you're going to give two bronchodilators, you may want to get a spirometry down the road to make sure you're not selecting people who will do poorly. Number two, from the same study is this very somewhat complex for those that are non-pulmonology slide that shows the second thing. Here is the response in FEV1, but no response in vital capacity. This is response in vital capacity and no response in FEV1, graded by the degree of obstruction. And what I want you to see is that people who have, or COPD patients, less obstruction respond more to the FEV1 with lower changes in the vital capacity. Whereas patients who are more severely obstructed do not meet the FEV1 response, but they have a higher vital capacity response. And this is very important because all trials I'm going to show you, all the data the FDA reviews is usually based on the FEV1 improvement. And on that basis, the more severe patients respond very little, but they deflate. And for the lungs, deflation is good. It is the preload of the ventilatory pump. All right. Let's see what happens. If that happens acutely, why don't we use it chronically? And now I'm going to go to Uplift and later on to Torch to show to you that if you then look over 4 years, you do not meet the goal of Uplift in the average patient. There was no difference between Uplift given tiotropium versus usual therapy on rate of decline of the FEV1. But if you look at the FEV1 per se after administration of the medication or even placebo, there was a benefit. There was a benefit in the number of exacerbations, and there was an improvement in the quality of life. So overall, on average, these patients will get an improvement in FEV1, no big change in rate of decline, but an improvement in outcomes. What was very interesting in this study is that a post hoc analysis of the grade 2, the milder patient, showed a difference in the rate of decline where that was not seen in the most severe patients. Now let's see what we were operating on. We only had 2 small cats, 4 times a day. This was the era of the high blood pressure, where is the doctor? When you use Aldomet, okay, 4 times a day. This is what we've been using for many years. They were identified by colors, a blue and a green. And we treated patients by colors. The pulmonologists treated patients by colors. However, the companies have turned the cats into lions, and they are ultra long-acting now beta agonists. I'm showing you some of them: indacaterol, olodaterol, vilanterol, this one that I don't even know what it is, and calmoterol. These guys stick to the beta receptor and they provoke a longer action by the LABA. And people have also developed the LAMA. For a while we had a LAMA, which was the ipratropium. Then came tiotropium that we labeled the 400-pound gorilla and made a big dent on being able to hang on to the antimuscarinic receptor. And now we have a jungle of gorillas: glycopyrronium, umeclidinium, and aclidinium. And they're all very good bronchodilators, pretty similar in shape, color, and distance. And I will show you papers that will deal with which one is better. Klaus Vogelmeier is here. He directed this study that I'm going to show you, where maybe this is beginning to look like the cardiologist curves. You get a slightly benefit when you compare a LAMA versus a LABA that was twice a day. So it would appear what we have all done, which is to give a LAMA once a day, every day, may be a little better than giving salmeterol twice a day. If you compare the ultra LABAs with the LAMAs, you will get benefits that are very similar in lung function. And you see them here for indacaterol versus tiotropium. So we may be reaching equality, parity, between the ultra LABAs that last once a day and the LAMAs that last once a day. Now the big question is 2 better than 1? And this is, you know, our cardiologists, brothers and sisters, have shown that 2 is better than 1. We are conservative, all right? I propose to you that, that perhaps if we evaluate them by some objective measurements, 2 may be better than 1, and if we combine them into one single monster, 2 are better than 1. And I'm going to show you a subsequent set of studies that do this. This is the first one of a series of studies published by Van Noord, where he puts the dromedary curves of either a LABA twice a day or a LAMA plus a LABA twice a day or once a day with a LABA. And this came out, and I thought this was very interesting. This is on the x-axis, time in hours, FVC on the y-axis. It could have been FEV1. If you give a LABA that lasts 12 hours, you get the dromedary. If you add the tiotropium only once a day, you get this line. But if you use the LAMA and the LABA, you actually get a bigger curve under the area, even if the short or 12-hour LABA was given once a day. So it would appear that by measurements of lung function, 2 is better than 1. And this has been repeated through all the molecules that you will see reaching your armamentarium. This is the GSK LAMA, salmeterol versus tiotropium or placebo, and you'll see plenty of these curves. You can read them easily. Placebo, even if it's German or English, doesn't do too much. But if you then use the single agents, it increases the FEV1. If you use the double agent, it gets a further increase. However, remember what I told you about some people not doing well on 2? This is not the same as a superior number of 1 on top of another. There is an improvement, but it's not as good as I would have dreamed, where you could double the value that you had at the baseline. Several of these studies have been done. I'm going to show you just a few, where if you compare 2 in 1 canister, this is one of the companies, QVA 149, compare it with the monocomponents with tiotropium, they sort of all bronchodilate 2 more than 1, but the difference is not as big as we would have liked. Having said that, the difference between 2 versus placebo is in every trial a lot more significant than the difference between 1 and the placebo. Now let me look at some other outcomes. What we have here is a change in FEV1 in this trial of umeclidinium plus vilanterol versus tiotropium and vilanterol in COPD. Again, an improvement when you gave 2 versus 1. However, when you look at the outcomes of TDI, a measure of dyspnea, side effects and exacerbation rate, the difference becomes smaller. There is always an advantage of the 2 over 1, but it seldom meets the minimally clinical important difference. And this holds true for almost every study that you look. I'm showing you here olodaterol plus tiotropium versus tiotropium plus placebo, 12-week studies, same change in area under the curve, 2 better than 1. SGRQ improved, but the difference was only about 2 units. So the theme on all these studies, and there will be many of them, there is not enough journals now. This is what's happening. We've been bombarded with all of these. We're running out of names. Instead, Illumina, Eulogio, Glow, Optimo, Lantern, Inspire. They're all short-term, Me Too, similar studies, 12 weeks, 26 weeks, and they all will give you the same message. So what do I conclude on my first part on bronchodilators? Combination of bronchodilators do bronchodilate more than monotherapy of LABA and LAMA. Combinations of bronchodilators impact on health status and exacerbations more than placebo. Invariably, they beat the MCID. However, the benefits of dual over single on patient-related outcomes and exacerbations is not as strong. Okay? Now let's turn to the anti-inflammatories, a big battle in COPD. I've depicted the anti-inflammatory as Tinkerbell. It decreases inflammation. So we bronchodilate and decrease inflammation is appealing. The one I'm going to concentrate is on ICS. I'm not going to talk too much about the other ones due to time. I think we have to remember that some of the results are not too dissimilar from Uplift. Torch had one big virtue. It targeted mortality, like our cardiology brothers have done, or sisters. Now, we didn't reach a mortality. It was 0.052. It could have been 0.048, and we would have said it reached it. I'm a doctor. I don't know if it reached it, but it did help a little. Huh? More so, if you look that there was a difference in the FEV1, there was a 25% reduction in exacerbations, and the quality of life was better. So the stool I have drawn is very similar than the one we have for Uplift. And on average, patients did get better. However, you paid a price. And that is that patients on this combination had a higher risk of pneumonia. There was no higher risk of death, but there was a higher risk of pneumonia. And as doctors, we should take this. Older, thinner, very severe, with many histories of infected sputum is not a good candidate for this combination, clinically. On average, this would be somebody you have to be very careful. Now, how does this have affected practice? What I did is I took all the large trials that have been done, including Wisdom, Helgo. And what happens is nothing has changed. Doctors are doing whatever the hell they want. Because there is somewhat of a confusion in the way we've positioned all these medications given the guidelines and the conflict of information that arises from so many products reaching the market at about the same time. I would like to go back to the beta agonist inhaled steroids to punctuate or at least bring out the idea that we perhaps have ways to move forward in this field that will select patients who may benefit versus those who may not. And I've chosen the glucose study because of the following. In the glucose study, they took 114 patients, not many. They had an FE1 of 60% because they were going to do bronchoscopies. They looked at FEV1 decline, SGRQ, and dyspnea. But what they did that I think is very important for us to learn is do biopsies, repeated biopsies of the lung to associate the outcomes with the pathobiology. Now, there were 4 groups here. I'm not going to bore you with all the different permutations. Just remember, one was placebo, the other one salmeterol, and the other one salmeterol fluticasone. And they did something that I think is extremely important, which is look at gene expression at 0 to 6 months, 0 to 30 months in placebo versus treatment. Now, I don't want you to read the lines or anything, but the patterns are different. And you know what? When you then looked and quantified the response of the FEV1, you will see that some patients actually improved, whereas some patients did not improve and some got actually worse. And when you look at the gene expressions of those that improved and did not improve, there were differences. Now, I cannot show you a periostin, an IL-6, a Th2. We are not there yet, but I do believe deep in my soul that some patients will respond to this therapy better and some will not respond well. There is a new kid in the block. This is Batman and Robin. You'll be hearing about Batman. I think he's showing up tomorrow. I think Ian is talking tomorrow. They've really pushed this cell, the eosinophil. This has become the hot ticket of asthma and COPD, and it does a bunch of things that are very interesting, but the normal range is between 1% and 3%. And we heard the question about the threshold values. I'm not going to talk more about this because tomorrow Batman is here. And I am just, Spider-Man, so I'm not as powerful. So I don't want to dwell too much in the eosinophil, but I believe we may have some insights in there as to who with COPD may do better on this combination. Are 3 better than 2? Why not? Well, there are some studies. This is tiotropium versus placebo, tiotropium salmeterol, or the combination, triple. And this has got one virtue. It was not done by a drug company. This was done by the Canadians in a combined trial. And what they looked was at exacerbations, patients with 1 liter, with only 400 patients, hospitalization, and there was no difference in overall exacerbations. But there were differences in hospitalizations, and there were differences that were significant in the FEV1. Now, I don't want you leaving this place and saying Celli said that everybody should be on triple. But the evidence is that if looking at some of these outcomes, as Tobias Welty did on this other study where he found a huge difference, triple versus single, in the rate of exacerbations at 3 months, it may very well be that the more severe patients benefit from triple, being careful on the side effects and selecting those patients on the fact that we are doctors, not robots. So what is my conclusion on ICS plus LABA? They are effective over placebo on lung function, health status, and exacerbations. May cause pneumonia. Be careful in older, thin, very severely obstructed patients with infected sputum. Finally, triple combination may be effective in patients with multiple exacerbations and admissions to the hospital, and perhaps the eosinophil. Now, I've been a doctor for 45 years. I graduated in May 1971. And I pride myself of being a doctor, not a robot. So guidelines are guidelines, but they're not straitjackets. And I think this becomes still an art. Medicine is the science and art of diagnosing and treating disease or injury and maintaining health. I think doctors should be giving some leeway when he sees a patient to interpret the personal issues of that patient and select therapies based on science, but also on his or her perceived feeling of how this will do on the patient. So how to approach? This is the poor doctor. ICS LABA, LAMA LAMA, LAMA LABA, SABA. PDE4 or SAPA. My goodness, then we come out with colors. We're running out of colors. Used to be blue, yellow, and green. Now it's the rainbow. Look at all the colors, canisters. If I'm a primary care person, I'd go crazy when the drugs reps, I run the other way. So I'm going to give you what Celli thinks he does, which is what I do. When my patient comes to see me, and I defended it yesterday in the Gold Committee, but I was overruled, I do spirometry. And I see if they're very symptomatic. If this thing looks like they're moderate, milder, I go like my brothers, the cardiologists. I give them lifestyle modification, smoking cessation, please do exercise, and I put them on a single, maybe a dual if they're more severe, but a single. And then I check adherence when they come again, and I consider ICS if no evidence of infected sputum. And I may go to triple therapy if things don't work well. If on the first visit the patient has severe COPD, very low FEV1, very symptomatic, New York Heart Association 3 or 4, I start with a dual, double dual, because whatever evidence I've shown you shows a bit more of a benefit. And I repeat the spirometry. I want to make sure their FEV1 doesn't drop when I give it to them. And if they don't do well, if they have been adherent, then I put them on the triple therapy. I go up to this. Now, there are specific phenotypes that I immediately look into. Deconditioning, rehab. The guy can, brush his hair, shave his face, or wash the hair if she's a woman. That patient needs rehab. Emphysema in the upper lobes, LVRS. If there's a lot of emphysema on the CT, I follow them for nodules in the lungs for lung cancer. I measure their oxygen, I walk them and see if they need oxygen. And finally, if they have a lot of exacerbations in spite of all of this, I may consider azithromycin. So my final conclusions, and you will forgive me if some of this was personal, healthy lifestyle, vaccinations, and exercise are a must for all patients with COPD. Pharmacotherapy, if symptomatic, we have the agents. I think combinations work very well if we monitor them. Finally, I think the future is ours to build. I think we need to put into the context of our thinking the possibility of using, since this disease kills 130,000 people, do trials like our brothers. No more of these 12 weeks, 24 weeks. We need trials like they've done. And I bet you that if we, with the tools we have, we put all our minds together, we'll be able to get the results they have. I think there is light at the end of the tunnel. Thank you very much.
Nico, and then Manolo. And Mario.
Thank you, Bart. You are great. But I'm coming from the last slides you show. Don't forgetting that we are doctors. I think what is your opinion about the growing, exponential growing of meta-analysis in the age of personalized medicine? So what is the value of this meta-analysis while we are going to the individual therapy?
Yeah, excellent question. Let me tell you what I think of meta-analysis. I call them data miners. People who don't want to do research with patients. They learn, they do computer research, and they go and put data in there, and blah, 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 and out comes data. The meta-analyses are very good if done by a very good meta-analyst who gets and selects the data very well. So if Gordon Guya shows me a meta-analysis, I tend to read it. But if a fourth-year fellow who never saw a patient in the 3 years he was there in the Harvard School of Public Health puts in and puts out a meta-analysis, it may confuse more than help. So some journals do not publish meta-analysis, some very good journals. They want original data. And we have many meta-analyses, systematic reviews that have led people to do things only to find out when a randomized control trial is done that the results were the opposite.
Thank you, Manuel.
Great, great lecture, and not only important but fun.
That's part of life.
One slide, the first slide, I think you showed that I think is very important, where you show the responses to bronchodilators in COPDers, and that you found out that there were a lot of 53% that responded to bronchodilators, but then you found another percentage that did not respond to bronchodilators. You went there then to show how the older people increase the vital capacity but do not increase the FEV1. In my experience, the main cause of a drop of FEV1 after bronchodilators is an increase in the vital capacity. So that, so I think we should, and then what is more interesting still is that even though we know that people with severe COPD will be volume responders, not FEV1 responders, we keep on looking at FEV1 and not at the inspiratory capacity. That is a different bird than the FEV1. In older people, so I think we have to somehow teach somebody, what physiology is all about and how does it work.
Dr. Chechi here, the cardiologist, is here or he left?
He's here.
He presented data which I loved, which is this molecule that decreases heart rate, a new molecule. Well, I think the problem with the very severe patient is that he's tachypnic, and he traps air almost during normal circumstances. And we very seldom measure the respiratory rate. Personally, I think our pump is just like the heart. It has a sinus node in the upper medulla. It has a conducting system, which is the phrenic nerve. It has a left ventricle, which is the diaphragm, a right ventricle, which is the accessory muscles, a preload, which is the volume, and a resistance, that is the airways dimension. We only look at the airways dimension. There isn't even one of the studies that I showed you where you can look at respiratory rate or lung volume. It is as if they had never looked at preload and had never looked at heart rate. You know, in my hospital or wherever I go, the respiratory rate is a constant, 18. Everybody has 18. They come in, they go to the ICU, they come. Actually, when they finish presenting, I go, what was the respiratory rate? Oh, 18. Yeah. So we go to the bedroom and I measure it, 26, 24. I wish I was younger again to do some of these things that in my
You are
mind.
young. Mario.
So I think we should measure volumes. That's the answer. Or short answer.
The problem that we are old. We graduated in the same year, just 2 months after you, me. It's like the reason why you don't remember what PF was, because it was a Pfizer beta-2 agonist, long-acting beta-2 agonist. We were in the board together in New York discussing with the people from FDA that were against this drug.
Okay.
That just to say.
Just to remind you.
Well, Nico has repeated the same question he asked me yesterday. We have just a meta-analysis. You know that I produce my own data.
I know
But
that.
he used meta-analysis because in this case, meta-analysis was useful, comparing all the 5 dual bronchodilation that we have today. And I'm sure that never we will have head-to-head comparison. In this case, meta-analysis is extremely useful to give at least some messages. One of the messages that we have drawn, and you will see the paper soon because they have sent me the proof, so I presume that it will be published very soon, is that always dual bronchodilation is much better than single monocomponent in the combination, because this is an important comparison that you must do, and not only on TRAF FEV1, but also on St. George Respiratory Questionnaire, although I don't trust St. George Respiratory Questionnaire, you know very well, and also about TDI. If you look at responder to non-responder odd ratio is 1.3. That means 30% more responders with the dual bronchodilation when compared with one single bronchodilator. Just to say what you can receive from meta-analysis. So,
Short.
Short. Furthermore, no, that's important.
Furthermore,
if you combine bronchodilators, mainly with the bilanterol and indacaterol, you can have a protection against severe adverse event cardiac outcome. That's an extremely important point. And meta-analysis in this
Mario,
case is extremely useful.
thank you.
What do you think about?
I think what I just said before.
Okay, thank you. Thank you.
That
thank
is not
you.
an answer.
Thank you.
We'll talk afterwards.
Thank you.